Dear learners, welcome you all in this course Systematics of Angiosperm. I am Dr. Gausia Bashri, Assistant Professor from the Department of Botany, Aligarh Muslim University, Aligarh. For this course, I am assisted by my PhD scholar Muhammad Subhan Ali, and now we are going to discuss about the evidences of cytology and phytochemistry in plant taxonomy. As we previously had discussed about the other evidences like evidences of anatomy, then we will study the palynology and embryology as evidence in taxonomy. Now we will going to study the evidences of cytology and phytochemistry. Then we will discuss some molecular tools that are also studied using for the taxonomic identification or as evidences. So moving forward to our content that we are going to study today, that is the cytology in relation to taxonomy, then phytochemistry, or we can say that the chemotaxonomy, then that is the ultra structure in relation to taxonomy, then genome analysis and nucleic acid evaluation in relation to taxonomy. So moving forward to our first topic for the today, that is the cytology in relation to taxonomy, that is the cytotaxonomy. As we have already discussed that the cytology is the study of morphology and physiology of a cell, and the information about the chromosome number, shape and pairing at the meiosis is used for the classification purpose. So cytotaxonomy refers to the ploidy levels and different types of ploidy type and for the chromosome aberration as data for the classifications. And then cytogenetics includes those studies dealing with the observation of chromosome pairing in behavior at meiosis. So if we see a chromosome, the normal structure of a chromosome is this particular. They have a centromere at the center. They have a two chromatid arm. One is a short, maybe one is a long, or they may be equal. Then this one is, if we see, this is a long arm and this one is the short arm. Then these are the termed as a secondary chrome section, and after that there is a satellite. So depending upon the position of centromere, chromosome may be metacentric, as the position of centromere is just near the center. Then submetacentric, where the chromosome is present side the one side, and necrocentric, as you can see here, it is just below the or near the short end, and in telocentric it is at the top of the arm. So these are some types of the chromosome depending on the position of centromere. And that further, if we study the chromosome, that some terminologies we have to know, like the first term, that is the karyotype, which is used for the phenotypic appearance of the somatic chromosomes, and the diagrammatic representation of this karyotype is termed as ideogram. So just the phonetic, if we represented them as a phonetic, then it is termed as the karyotype, and if we represent them as a diagrammatic, then it is termed as the ideogram. And then in taxonomy, the characteristic chromosome which have proved to be taxonomic value includes the, as we have told you, the chromosome number, chromosome size, morphology and its behavior. These are the main characteristics which is useful in the taxonomic aids. Now then, moving towards the chromosome number. This is as it is the number of chromosome in each single cell. So we are generally aware that the number of chromosome in each cell of all the individual of a single species is constant. Suppose like we are the humans, we have a 46 number of chromosome and 23 pairs, we can say. There is a n is a 23 and we have 46 number of chromosome, and in the pair we can say 23 pairs of chromosome in each cell of our body. So it is also established that the more closely related species are likely to have a similar chromosome number, while the more distant related ones shall have a different numbers of chromosome. So due to this relative conservation, chromosome numbers become an important and frequent use for the taxonomic characters. So in addition to this, there is a very wide range of chromosome numbers in the angiosperm, from low as they can vary from the 4 in case of Haplopipus gracilis in the which belong to the family Astraceae, to as high to the 530 as in case of a Poa litrosa that belong to the family Poaceae. So there is a, you can see there is a large number of version in the chromosome number from 4 to 530. So this is a large number of angiosperm has been analyzed for their chromosome numbers that provide a good information for the taxonomy. So many interesting ideas have been developed due to the knowledge of these chromosome numbers. As you can see, there is a different species generally have different numbers of chromosome. You can say that is the Haplopappus that has a 4, Pova, then Calonche have a 500 number, then Ophioglossum which is a pteridophyte that has a 1240 number of chromosomes. So you can see in the plants there is a quite number of variation in the chromosome numbers, and just in angiosperm it varies from 54 to 530, and this one is a pteridophyte. So there is a some correlation in these numbers, as if we see many interesting ideas have developed from the knowledge of this chromosome. For example, in the genus Festuca, the different species of different chromosome number forming a mathematical series, as you can see here. In the different species, they have a 2n number varying from 14, 28, 42, 56 and 70, etc. From this information, a generalized can be made, that is different species maybe have a some common basis. If we assume that these are the chromosome numbers are used as a common denominator called x, that is the 7, then we can consider the species to have a multiples of the number. So we can see that the denominator of base 7 then can be considered for the basics set of genetic information carried by a plant, and due to the multiplication of this basis genetic set, the evolution of different species has occurred. So as a series, we can see such a series said to be polyploid, in which the basic number x is equivalent to the haploid number of chromosome in a diploid series. So if we see the different species, then if 4x, then it is called as the tetraploid. Then if it has a 6x, then it is called as the hexaploid. Octaploid, if it is in comparison to this basic number, then it is called as octaploid. Then decaploid if it has a 10x respectively. So it is forming a series, and we can predict the ploidy depending upon the species to species. So similarly, if we see the chromosome number, the many plants are the homoploids like Pinus and Quercus, they have a chromosome number 12. Then polyploids like different species of aster have a chromosome number 9 or 18 or 27. As we can see, there is also series like 9, there is a in the denominator of 9 all the species of this aster. Then euploidy, as euploidy the different species of Brassica bears like 6, 7, 8, 9 or 10. That is the aneuploidy, there is an addition or deletion in the number of chromosome. So polyploid has a multiples, and here the number of chromosome is increasing by just 1. So there are some common trends we can see here in these chromosome, different groups of chromosome ploidy. And the next character that is also may be important in this is the chromosome size. The chromosome sizes vary from 0.5 to 3 micron. Then according to Stebbins, the chromosome size is a characteristic of the certain groups and families and not related to phylogeny or angiosperms. And the next important trait in the cytotaxonomy is the chromosome morphology. Like characters like, the next important attributes in the cytotaxonomy is the chromosome morphology. Characters like position of centromere, as we have discussed earlier, the arm and the length of the presence or absence of knob have been considered for the taxonomic purposes. Like chromosome also vary from size, volume and in amount of distribution of heterochromatin. Like most of the chromosome are V-shaped, and in advanced genera, chromosomes are generally J-shaped. As here you can see the chromosomal structure, that is the acrocentric, this B1 is the metacentric and the C is the telocentric. Then depending upon the appearance of the basic chromosome cell in a dividing cell is known as the karyotype of the cell, and this can be analyzed to provide the information not only about the chromosome number but also about the chromosome size, volume and type of the chromosome in the cell. So this information is used for the taxonomists to identify plant and understand text relationships, and the karyotype can be represented diagrammatically as an endogram or karyogram, and these diagrams can be compared for the taxonomic purposes. Like another interesting observation in this, that the absolute size of the chromosome of a karyotype is fairly constant since it is controlled by its genotype. So the taxonomists have found that monocots generally have a larger chromosomes than dicots, and that smaller chromosomes are found in hardwood plants in comparison to their herbaceous relatives. So there are some trends in chromosome numbers, as if we see the Peonia was the earlier placed in the family Ranunculaceae. However, on the basis of their embryological and other evidence, it is suggested that it placed in the independent family Peonaceae. So, cytologically, it is found that the Peonia possess large chromosome as opposed to the smaller chromosome that are the characteristics of member family Ranunculaceae. So, because of this, it is now removed from the Ranunculaceae and now placed in the family Peonaceae. Then, the plants of Polygonum that growing in 3 different habitats exhibited morphological differences and were hence considered belonging to different species. But interestingly, it was found that all of them were not only interfertile but had similar chromosome morphology. Hence, Polygonum species is now clubbed into. So, there may be some species are removed from particular genre, or they may be clubbed into this chromosome morphology. Similarly, the chromosome character studies have suggested that placing the genus Butamus in a separate family Butamaceae while retaining the Limocaris, Hydrocaris and Tenagocaris in the family Alemastacea. Then, the closer affinities of the family Cyperaceae and Juncaceae have been established due to the presence of a holocentric chromosome in their members. As we've already previously studied, the embryology of the Cyperaceae and Juncaceae is also somewhat similar. As we can see here, the chromosome morphology of these two families are also quite similar. So, we can say that these two families are quite have a same ancestral origin, that Cyperaceae originated from the Juncaceae family. Then, if you see the karyotype studies have come from the separation of the family Agavaceae from the Emeraldaceae, and the presence of 5 large and 25 small chromosomes in their cells have helped the transfer of Yucca from the family Liliaceae to the family Agavaceae. Then, if you see the chromosome behavior at meiosis, and when we study the meiosis, we not only observe the regularity of pairing, which is important for the fertility of the plants, we are also able to make a chromosome to comparison. This provides a valuable information about the role of chromosome in heredity and the taxonomic species of plants. So, we can also determine the nature of the genome to find out its homozygous or heterozygous. Then we can also study a very significant study to this in this regard is the case of common hexaploid breadwheat, that is the Triticum aestivum. The genome of this economically important plant has been designated as AA, BB and DD with 2n is equal to 42 chromosome. When detailed genome analysis have been established, that 3 different species have contributed to the evolution of this hexaploid wheat, and the genome A is from the wild grass, that is the Aegilops speltoides, that is at 2n number is equal to 14, and the D genome has been contributed by the Aegilops sclerosa, that has also chromosome number 2n number is 14, and many geneticists have suggested different grasses at the source of the B genome. And then if we see the meiotic abnormalities such as non-pairing of chromosomes, crossing over, lagging chromosomes, bridge formation, non-disjunction and translocation can be easily detected and analyzed for taxonomic studies. Such differences in chromosomes behavior have been noticed in different species like in Datura stramonium. So here they provide a sort of reproductive isolation barrier required for the ecological differentiation of the population. And depending upon the chromosome binding patterns, according to Horn, chromosome binding pattern reveals structural heterogeneity and can shed light on the some structural and functional properties of the chromosome. And these differential binding patterns help the taxonomists in identifying a homologous chromosomes, the form groups of resembling karyotypes, and the separation of different karyotypes. So, this binding pattern is very important in this following identifications. And then the next is the systematic value of cytological studies, that we have seen that the many groups are important for the. Now we will see how it is important, these cytological studies important for the different identification of different types of families. Like I have already told you, the member of Cypresse and Junkesi possess chromosome with diffuse and non-localized centromere and also show inverted meiosis. This reflects a close association between these two families, and that's why they are now considered as a very close to each other. Then Yakka had a long been treated as a member of Liliaceae, but because of the superior ovary and agave from of Amaryldaceae because of this inferior ovary. But Hutchinson shifted both the plants to Agaveaceae because of the presence of 25 small and 5 large chromosomes in both of them. Because this Agaveaceae family is a 25 small and 5 large chromosomes. Due to this, these two plants, Jacca and Agave, shifted to the Amaryldaceae by the Hutchinson. Then the basically, then moving forward to the some other examples of cytological studies in the taxonomy is the like the basic chromosome number in Lorenthaceae is 9, while in Viscaceae there is a series of aneuploid number ranging between the 10 and 14. So we separated them from each other on the basis of cytological evidences because Lorenthaceae has a basic number that is 9, while the Viscaceae has a aneuploid that is ranged between the 10 to 14 as we have already seen in the examples. So in the family, similarly Bambusoideae of Gramineae, the n is 12 and in the subfamily Purdy the n number is 7. This indicates that the chromosome number have proved to be taxonomic utility and also at the subfamily level. Then Stebbins provided information on the evolution of grasses on the basis of cytogenetics. Then on the basis of cytological studies, Lewis submerged the genus Godelschia in Clarkia in the Onagraceae. Then Naik differentiated 3 species of Chlorophytum of Liliaceae on the basis of cytological data. Then according to him, the Chlorophytum Barochi has 2n is equal to 16, while Chlorophyton glaucon and Chlorophyton glaucoides have 2n is equal to 42. Both the later species have different karyomorphology. So they have differentiated these 3 species of Charophyton. Similarly, Warburg studied taxonomy of Naik differentiated 3 species of Chlorophyton of Liliaceae on the basis of cytological data. According to him, Chlorophyton barochii has 2n number is equal to 16, while Chlorophytum glaucum and Chlorophytum glaucoides have a 2n number is equal to 42. So both of the later species having different karyomorphology, that's why they have a differentiate 3 into the 3 different species. Similarly, Menton confirmed the formation of a subdivision Brassicaceae on the basis of cytological studies because all the families have a different base of chromosome numbers. So they subdivided the Brassicaceae family into different groups. Then genus Citrus that belong to the Cystaceae formerly included in the Helianthemum as chromosome number 8, while Helianthemum has base chromosome number 9. So Citrus. Then the genus Cistus that belong to the Cystaceae family formerly included in the Helianthemum, which has a chromosome base number is 8. Then genus Cistus which belong to the family Cystaceae formerly included in the Helianthemum and has a chromosome base number that is the 8, while Helianthemum which has a chromosome base number is 9. So Cistus should be recognized as a separate genus. Now it is confirmed. The new classification of the genus Narcissus of the Amaryldaceae has been proposed by Fernandez on the basis of their cytological studies. Then Sharma on the basis of his studies of Araceae, Amaryldaceae and Dorseraceae family proposed that the changes in karyotypes of somatic tissues play a distinct role in evolution. He further proposed that the large chromosomes and low chromosome number and symmetrical karyotype represent a primitive status, while small chromosomes, high chromosome number and extreme symmetry and karyotype represent a advanced status. So, due to presence of these characters, we can see which species is advanced and which species is primitive. So, these principles provide interesting result in taxonomy of alismestaceae, liliaceae, emeraldaceae and dioscoraceae. Now moving towards the next topic, that is the phytochemistry in relation to taxonomy. So the science of chemical taxonomy is based on the classification of plants on the basis of their chemical constituent related with their molecular characteristics. These principles provide interesting result in taxonomy of alismataceae, liliaceae, emeraldaceae and dioscoraceae. So these are the some cytological studies that help in identifying the different families. As we have seen, many families are recognized and genus on the basis of their cytological studies. So as we can see, this is a very important evidences in the taxonomy, that is the cytology. Now moving towards our next evidence, that is the phytochemistry in relation to taxonomy. It is also called as a chemotaxonomy. Like the science of chemical taxonomy, that is based on the classification of plants on the basis of their chemical constituent related with the molecular characteristics. So we can say that application of chemistry to taxonomy is called as chemotaxonomy, and the chemical evidences is used to determine the relationship among taxa of different categories. So some of the major classes of chemical evidence include flavonoids, alkaloids, amino acids, fatty acids, aromatic compound, terpenoids, polysaccharides and carotenoids. And it also includes the investigation of patterns of the compound existing in plants, and then investigation of pattern of the compound in plants parts like bark, wood, leaves, roots, etc. Then this popularity of the phytochemistry or chemotaxonomy is due to the development of rapid analytical techniques. The belief that data from the many sources should be employed for the classification. So all the branches now come together and we have now analyzing all the different aspects onto the one side in the taxonomy. So if we see the phytochemistry in relation to taxonomy, it is, as I have already told you, that is the science of chemical taxonomy. It is based on the classification of plants or on the basis of their chemical constituents. So if we see the chemotaxonomy, like the plant produces several kinds of natural products in varying amount and quite often according to their own biosynthetic pathways. These compounds also differ from one taxonomic to another taxonomic groups, and the occurrence, distribution and biosynthetic pathway of various types of chemical compound present in plants prove to be taxonomic significance. And this chemotaxonomy is based assumption that similar or related groups of plants have similar chemical compounds. Thus, the plant chemicals play a key role in identification of the systematic botany. So, it gives a close relationship between chemical constituents of plants and their taxonomic status. That's why it is very important to analyze the different chemicals present in different groups of plants. And this chemotaxonomy, according to Menzer, recognized three principal kinds of chemical constituents that helpful in taxonomy. The first group in this is the basics or primary constituent, that is protein, nucleic acids, chlorophyll and polysaccharides. Then the second group is secondary constituent, that lacking nitrogen and not involved directly in the plant metabolism, like simple phenolic compounds, then like flavonoids, terpenes, coumarins, alkaloids and many others miscellaneous substances, like their use in taxonomy is not developed so far. So if we see on the basis of chemical, as we can see, these are on the basis of their cycle and their use. The second group of classification of this compound is depending upon their size. Like on the basis of their molecular weight, Jones and Leschinger have divided the natural chemical plant products useful in taxonomy into two major groups. The first one is the micromolecules and the next one is the macromolecules. As the name indicates, micromolecules are compounds of relatively low molecular weight, that is the 1000 or less. And the example of these are the amino acids, alkaloids, cyanogenic glycolides, fatty acids, terpenoids, flavonoids, anthocyanin and betalains. While the macromolecules are high molecular weight compounds over 1000 molecular weight, examples proteins, DNA, RNA, complex polysaccharides. So this is just on the basis of their molecular weight. If we see one by one of these particular compound, like the first one is the tannins. These are the amorphous substances with wide distribution pattern in plant kingdom. Tannins are known for their astringent flavor and form colored solution with iron and other metals. And if we see like Anacardiaceae, Lauraceae, Punicaceae, Magnoliaceae, Miliaceae, Moraceae, Sapindaceae, Septaceae, Rosaceae, Utricaceae, Thiaceae are tanniferous families. They all have a tannins. And Acanthaceae, Amaranthaceae, Campanillaceae, Chenopodiaceae, Cucurbitaceae, Lamiaceae, Solanaceae, etc. are the non-tanniferous families. So we can just categorize these two groups. First, that have a tannins, that called as the tanniferous, while those groups of the families that don't have a tannin are called as the non-tanniferous. So this is just a broad category of classification depending upon the presence of tannins or absence of tannins. Similarly, steroids, the next group of a compound that present in the plants are the antisteroids, may be considered as a derivative of fused and fully saturated drink system called as the cyclopentophenylnephrine or styrene. And these are the cordinolides or corditonic glucose found in the family Apocynaceae, Asclepiadaceae, then Celastraceae, Euphorbiaceae, Scrofulaceae, Ranunculaceae, Rosaceae and Salicaceae. So you can see the presence of steroid in many groups. As we can know, the Apocynaceae and Asclepiadaceae, these families have a quite. The next group of compound is steroids. Steroids may be considered as a derivatives of a fused and fully saturated drink system called as styrene. These are cardinolides or carditonic glucosides found in the family of Apocynaceae, Asclepiadaceae, Celastraceae, Euphorbiaceae as in case of Molletus, Scrofulaceae as in case of a digitalis, Ranunculaceae as in case of Isoplexis, Rosaceae in case of a Malex, and Salicaceae in case of a Salix. These are some groups which contain the steroids in plants families. Then the next group of plant compound is the alkaloids. Alkaloids are heterogeneous group of nitrogen containing organic compounds with nitrogen containing heterocyclic nucleic acid derivatives from a biogenic amine and is structurally related to the parent base such as the pyridine, piperidine, isoquinone and tropane. So alkaloids distribution has been proven useful in taxonomic tool in plant systematic. Like for example, alkaloids protopine, which is found in the all species of the family Papaver and Fumaraceae, thus raising the close relationship between the Papaver and Fumaraceae to the presence of the alkaloid protopine. But all the members of the family Fumaraceae have a combination of isocanine derivative, while these are absent in the members of Papaver. So we can see these are the closely related but also a distinct group of families. Similarly, Barbidaceae, Fabaceae, Ranunculaceae, Solanaceae are rich in alkaloids containing families. The next group of the compound, that is the phenolics and flavonoids. Flavonoids are the largest group of naturally occurring phenols and mostly found in the vacuoles of the plant cells. Like phenols contain hydroxyl group directly attached to the aromatic nucleus, and the flavonoids include the flavones, flavonones, isoflavonones and anthocyanins. These are some groups of compound that present in the different groups of families. Then the next compound that is present in the different families and very important, that is the terpenoids. These are the volatile compounds responsible for the essence in plants like mints, citrus, umbelliferous, gymnoids. Then the next group of the plants that is responsible for the then the next category of compound like terpenoids. These are the volatile compounds responsible for the essence in plants like in mints, citrus, umbelliferous and gymnospermous, etc. Terpenoids are mostly polymerized isoprene units that is made up of 5 carbon, and on the basis of their isoprene units in terpenoids molecules, they are categorized as the followers. Like monoterpenoids, those have 2 isoprene units like menthol. Then sesquiterpenoids, those have 3 isoprene units like in case of a farnesol. Then diterpenoids that has a 4 isoprene units is called as the phytol. Then triterpenoid that have a 6 isoprene unit that is called as the, in case of example like squalene. Then tetraterpenoid that have a 8 isoprene unit like example beta carotene. Then polyterpenoid that have a multiple isoprene unit and like example latex and rubbers. So these terpenoids are almost universal, although not as a widespread as flavonoid, and have been used extensively in the chemotaxonomy of mint, umbelliferous, citrus plants and gymnosperm. Some specific terpenoids like are the characteristic of particular families, example lactones in many family of lactones in family Asteraceae, cucurbitacins in Cucurbitaceae and aspergilloside in case of a Rubiaceae. So as previously we have seen that the flavonoids are very common, but terpenoids generally they occur in a specific group of families. So they can also act as a very good taxonomic tool in identifying different types of families. Then the next compound, that is the betalains. Betalains are the pigments that have a different from flavonoid and other phenolic compound due to the presence of nitrogen in them. However, they are functionally equivalent to phenolics, popularly known as the nitrogenous anthocyanins. So red to violet beta-cyanins and yellow beta-xanthines are confined to 10 families of angiosperm which contain only betaline but no anthocyanin, like Chenopodiaceae, Portilocaceae, Isosaceae, Cactaceae, Nyctaginaceae, Phytolocaceae, Stegno, Spermaceae, Basilaceae, Amarantaceae and Dideraceae, which have been included under the single order Centrosperming. So you can see here, just because of the presence of these beta lins, these are the 10 group of families that are confined to the single order, that is the Centrosperming. Then the next group of compound is that is the iridoids. Iridoids are the separate class of monoterpenoids, cyclopentanoid lactones, which also represent a separate class of taxonomically significant compound. And the distribution of this iridoid compound has been proved helpful in tracing the phylogenetic ancestry. Like several isoid compounds like aspergilloside are particularly common in the family Rubiaceae, and like aucubin is frequently found in the family Cornaceae, Scrophulaceae, Orobanchaceae, etc. And the aucubin containing genus Budeja transfer from the Logoniaceae to the Buldaceae with position near to the Scrophulaceae due to the presence of this particular compound aucubin. So you can see here further this presence of this particular compound is reason to become the support of evidence that is a transfer for the Budeja from this particular Logoniaceae to the Buldaceae for the near Scrophulaceae. Then here the some cytology. Then the next group of compound is the cyanogenic compound. The poisonous compound produced by the plants after injury to the cells are called as cyanogenic compound, and such plants are called as the cyanophoric plants. The examples of such compound are hydrocyanic acid, then amyglidine, etc. And the cyanophoric plants usually contain one or several cyanogenic glycols, like the taxa belong to the family Araceae, Poaceae, Juncaceae, Juncaginaceae and Schuzaldaceae are the common cyanogenic plants, and the several dicotyledon families belonging to the family Astridae, Rosidae, Dilnidae are also cyanogenic. Thus, the use of, we can say that the use of the chemical criteria proved to be an efficient taxonomic tool in the field of plant taxonomy or systematic, because on the basis of this we can identify different groups of the families, because we can see here the different some group of the plant are present and chemicals in particular group of the plants. That is the basis of the taxonomic to differentiate them. Then as we have seen the micromolecules and macromolecules, and then the next group of plant, that is the next group of compound, that is the cementitides. The cementitides are those compound or a group of plant that carry some information, like these are the information carrying molecules in plants are called cementitides, and they have been recognized to be a three kind, like deoxyribonucleic acid or DNA, that is the primary cementicide, then ribonucleic acid or RNA, which is called as the secondary cementide, and the protein that is called as the tertiary cementide. Following the sequential transfer of the genetic code, as we know, all the information, genetic information of our stored in DNA. From DNA, it is transcripted to the RNA. Then after the translation, is now going to the protein. So of these, the protein are the most favored molecule for the chemotaxonomic purposes, because the plant protein can be studied by different methods, by electrophoresis or by serological method, and both processes have been used for the obtaining information about the protein chemistry of different plants. So these cementides are very important in identification of different groups of the plants. And if we see the common breadwheat, that is the Triticum aestivum, the storage protein will analyzed by electrophoresis, and for comparative purpose, the storage protein of the tetraploid wheat, that is the Triticum dicoccum, and the diploid grass Aeglocopus scarosa were also analyzed electrophoretically. And this study confirmed the conclusion that the hexaploid wheat did contain a sum of protein possessed by the diploid species, which have contributed to the evolution of this hexaploid wheat variety. So it is confirmed that this particular wheat varieties come from this Triticum dicoccum and this diploid grass, that is the Aeglocopus scarosa. So this study support the observation based on the morphology and the cytological evidences, as we have already seen the previous slides of the cytology on the evolution of Triticum aestivum. Then if we have to study the further these proteins, these are the some method called the serological analysis of protein. So this serological analysis of protein is based on the immunological reaction shown by mammals when a foreign protein is introduced into the system. In other words, this is the based on the antibody and antigen interaction, where the antibody is being specific to an antigen bringing about the coagulation. So this information can be then analyzed to understand the relationship of the different plants on the basis of their serological evolution of the plant protein. So serology has proved to be a useful taxonomic tool as different levels of classification. And J.G. Hawkes and his co-workers studied several tuber producing species of the Solanum to understand the evolution of cultivated potato, that is the Solanum tuberosum, and determine the species of Solanum which could be established as the ancestor of the common cultivated potato. And so we can see that a general conclusion from such studies in that the different amount of serological activity in members of different plant families may be interpreted as a reflection of evolutionary differences in the primary structure of protein, due to the which serological differences can be recognized between the members of different families. So it is a very important step to analyze the evolutionary history of ancestrals. Then the next we will study about the ultrastructure in relation to taxonomy. As we have already seen, our studies different evidences from morphology to phytochemistry or chemotaxonomy. Now ultrastructure is also very important, because after the discovery of electron microscope studies, there are several new advancements in the group of plant taxonomy. So electron microscope studies have also played an important role in taxonomy. That is, Hayward and Dakshina demonstrated the use of electron microscopy in taxonomy. And the important characters used in classification are as follows, like the sieve tube plastid. Then Becke studied more than 1500 species belonging to 380 families for sieve tube plastid, and found three types of plastid depending upon the accumulation of starch and protein, that is the S type, SO type and P type. So these are the three types of plastids, and then they also categorize detailed cisternae, and that is termed as the DC. The inversion effort found that the dilate cisternae in the capillaries and DC in the endoplasmic reticulum were reported by Bonnet and Newcomb in the root cells of Raphanus sativus. Then Favalli and Garola reported DC in the phloem parenchyma of the foliar veins in Brassica chinensis, and Jorgensen found them in the capillaris sieve elements in the evolutionary origin. So these are the some ultrastructure studies that supported the taxonomy. Then The next is the genome analysis and nucleic acid hybridization in relation to taxonomy. Genome analysis and nucleic acid hybridization contain identification, measurement and arrangement of genetic features. These genome analyses have very crucial role in understanding genomic relationship among species and it is important for systematics, evolutionists and molecular biologists. Like genomic relationship among the diploid species, like genomic relationship among diploid is determined as the crossing affinity, like interspecific crosses involving parental species with similar genus usually set normal pods and seeds, while crosses between the dissimilar species seeds are commonly abortive. Then the genomic relationship among diploid species. It is very important to determine the affinities between the genetic and genomic species of diploid, and among these the first one is the crossing affinity. Like interspecific crosses involving parental species with similar genus usually set normal pods and seeds with crosses between dissimilar species, then the seeds are commonly abortive. So some hybrids do not set seeds and something the crossing is successful in one direction and it is called as genotype independent. Then similarly, if we can also study it by the chromosome pairing. In interspecific hybrid, the degree of chromosome pairing facilitates the analysis of phylogenetic relationship among the species, providing information about ancestral types. Then, depending upon the DNA-based molecular markers are applied in the various aspects of taxonomy to analyze the genetic identity, genetic relatedness among the population, geographical population and species, their pedigree, and the differentiation among the isolated species, and phylogenetic structure at various micro and macro levels. So, if we have to study these phylogenetic studies, that we have to see the molecular data. So, in the preceding section, we have already learned about the nucleic acid and protein constituent that is called as the cementioids. However, only nucleic acid, that is the DNA and RNA, contribute toward the significant analysis of phylogenetic relationship among the plants. And traditionally, the taxonomists use tool that is used for the taxonomy to construct phylogeny of the plant or plant groups. So, however, has now been realized that the molecular data based on the version in DNA and RNA of different plants provide more reliable input, and such an assumption is based on the hypothesis that the phylogenetic closer species should possess similar genetic material and distant species should exhibit variation in their genetic constituent. So, in recent times, there is a technique that is called as the DNA fingerprinting has become very useful source for information in taxonomy. It allows comparison of different species of genus, enabling understanding of phylogenetic relationship. So, for this technique, by using this, we can assess how the two species are closely related with each of them or how far they are closely related. So, our understanding of cell biology reveals that such molecular data lie within the cell organelles, nucleus, mitochondria and chloroplast of the cell. So, all of them possess DNA and are referred to as a nuclear DNA, mitochondrial DNA and the chloroplast DNA respectively. So, nuclear DNA of a cell generally consists of a million to many billions kilobase pairs and is biparental in origin as a combination of male and female genome. Though the stable, it is evolved rapidly and more difficult to analyze. However, it is a source of data that could throw light on the final evolutionary process of a particular organism. So, if we study both mitochondria and chloroplast DNA, they are uniparental in origin, and based on the observation that mitochondria DNA is made up of 200 to 2500 kilobase pair, undergo a lot of rearrangement as well as the occurrence of many forms of it within a mitochondria. So, it is regarded as relatively as of little significance in the phylogenetic systematics. But the most suitable genetic material, the taxonomic evidence of green plants is that they are chloroplast DNA. Because of this, in higher plant, the size of chloroplast DNA in chloroplast is only 120 to 140 kilobase pair. Thus, it is the smallest among all the three sources of genetic material within a cell. So, this is the advantage of studying the chloroplast DNA in taxonomic study. It could be summarized as follows. Because of their smaller size, the large number of chloroplast per cell, so it is easily to study their relative stability on not only in organism but also in a species. Then easy to isolate and analyze, then not much altered by the evolutionary process, and all the chloroplast DNA molecules carry basically the set of genes. However, arrangement varies in different species. So this is the most suitable type of DNA material, that is the chloroplast DNA, to study frequently for the taxonomic evidences. So we can say that the molecular data is being used to construct the phylogenetic tree of the green plant at almost all ranks. So principally it helps in the establishing affinities and relationship within the and outside the ranks. For example, that is the monophyly of the families Apiaceae, Caprifloraceae, Pentagenaceae, Scrophulaceae, Apocynaceae and Saxifraceae etc. is well supported by the molecular data analysis. Further, it also supports the union of families like Apocynaceae and Asclepiadaceae and the Brelliaceae and Apiaceae and Brassicaceae and Caparaceae. So these families are very closely related to each other. That's why this relation of molecular data reveals the association of this union of these particular groups of families. So we can say that by the studying this particular study, for the proper characterization, identification and the classification of the plants, these taxonomic evidences from a variety of sources should be gathered prior to any definite interpretations regarding phylogenetic relationship among various plants or plant groups. Because if we only see a one particular characteristic, we cannot conclude that this is the group of that particular plant. We have to analyze each and every aspect of this evidences. Then we have to analyze them, and then we have to decide what is the particular group of that particular plant, which is very important to assess all the evidences that available for that particular plant, from its morphology to its molecular data. That's why there's a now taxonomic ranges is starting from the morphological studies to the natural, and we have to incorporate all of them. That's why it is termed as the omega taxonomy, because we are now studying all the aspects and then we are combining all of them. Then particularly we classify or identify a group of plant on the basis of this all the characteristics. So this is all for the evidences. Thank you all of you for joining this session. Thank you all.